Hello student, this is Miss D.A. Malvekar working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. And in this video, we are going to discuss about development of animals. In that, just we will first look upon the structure of a gamete. As we know, if we consider the human being, then the sexes are separate. That is, the male individual and the female individual, we can distinguish them according to their secondary sexual characters, as well as if we study their reproductive organ, then reproductive organs are also differ in the male and in the female body. So what is the main function of this reproductive system? This reproductive system will generate the gamete. Suppose if the body is of the female, then female body will produce the female gamete that is called as a egg. And if that body is of the male individual, then it can synthesize the male gamete or which is also called as a sperm cell. Now just we will quickly see what is the structure of a male gamete. Obviously, it is a smaller in structure as compared to the female gamete. When it is developed in the male gonad, it is synthesized or it is produced in the millions of copies. It is motile one because it is having a flagella. It is having divided into major four parts, that is head, mid piece and tail with the end piece. The head is actually it is developed in such a way that it can occupy the haploid cells coming from the gamete. And obviously, the cytoplasmic matrix is very reduced at the head. At the proximal end of the head, there is a acrosome which is developed from the Golgi bodies. And the main function of this acrosome is when this male gamete will fertilize the female gamete, then this acrosomal breakout will release certain enzymes which will release or it can reveal the structure between the or the cementing structure which is present in the follicle cell during fertilization. In the middle piece, the number of mitochondria are circled around here. And the main function of a mitochondria is it is giving the energy for the movement of a male gamete. And the tail is just for the motility. Now, if we consider the female gamete, it is having its own haploid set of a nucleus. It consists of its own cytoplasm, and the outermost covering will be the zona pellucida is the transparent covering present around the egg. First covering is of the cell membrane itself, which is surrounded by the thin transparent covering which is called as the zona pellucida. And the zona pellucida is surrounded by the vitelline membrane. This vitelline membrane after fertilization is converted into the fertilization membrane. And after that, you will observe here that these are the number of cells which are surrounding to the egg, and that cells are called as a follicle cell or nurse cell. And if we observe it in the diagram, then it is obviously we can visualize it as a corona of the sun. So this cell or this layer is called as a corona radiata. Now, if we consider the process of gametogenesis, the first and important process in the individual is formation of gametes. And this process of formation of gametes is called as a gametogenesis. Now, as the individuals are separate or as the sexes are separate, if the individual is producing the sperms or the process of formation of a sperms is called as a spermatogenesis and process of formation of a egg is called as a oogenesis. Now, if we consider the process of oogenesis and the spermatogenesis, the major difference is the spermatogenesis takes place in the male gonad, that is testis, and the oogenesis will take place in the female gonad, that is ovary. The process is near about the same, but it is not identical because gamete formation requires the process of a meiosis in which The one cell will divide and reductional division takes place to produce the four haploid cells. So similarly, here also the sperm mother cell is called as a spermatogonium, and there are number of sperm mother cell are present in the testis. Now, if we consider the single sperm mother cell, it will divide and it will start to proliferate. It will maintain its own copy, right? And in its life cycle, it is having a three different phases. First is a proliferation or the multiplication, then it is a growth, and then it is a maturation stage. So during multiplication, it will try to form the number of cells which are looking alike by the simple process of a mitosis. Once it will finish it will increase in the size, and it will get entered into the growth phase, and after growth phase it will become the mature in this serially as shown in the picture. So once the growth will occur, this sperm mother cell will be converted into the primary spermatocyte. Site is the cell. Then this primary spermatocyte will undergo the meiotic first division, and it will converted into the two cells, and that cells are called as a secondary spermatocyte. And this secondary spermatocyte now able to complete this meiotic division, and they will form the four haploid sets that is called as a spermatid. Now if we observe this spermatid, it is not looking like a sperm cell which we have studied here. That is, it will be having a head, middle piece, and the tail. It is generally during gametogenesis, the spermatids are formed by the process of meiosis. But when these spermatids are formed, they will get differentiate in the testis itself, and they will form the acrosomal body, then the head containing the haploid set of a nucleus, then the middle piece consists of the mitochondria, and the tail. So the fourth spermatid will give the four sperm cells. So this is for the just one spermatogonium. So number of sperm mother cells are there, and they can form the million copies of a male gamete into the male gonad. Similarly, as compared to this one, the process of formation of an egg is called as the oogenesis, and which will take place in the ovary of the female body. Now, similar to the spermatogonium, here the egg mother cell is called as a oogonium. Number of egg mother cells are called as a oogonia. Here also it will undergo the mitosis process, and it will maintain its own copy. And then in the growth phase, it will be converted into the primary oocyte. Now there is a difference between this step in the spermatogenesis and in the oogenesis. As the spermatogenesis, it will proceed further, but in case of oogenesis, once the primary oocyte is formed, it will remain as it is, and the meiosis process will be arrested at this phase in the baby girl child. But when that baby will become mature, then it will this arrested meiosis will become completed in her reproductive phase when she will become mature. And then this meiosis 1, which is arrested at earlier stage of development, it will complete its division, but it is an unequal division, and which is called as a one. Say it will divide unequally, it will form the larger cell and the smaller one. The smaller one is called as a polar body because it will attach to the other end of this cell, and another bigger one which is called as a secondary oocyte. Now here also it is get arrested, and then during puberty stage, this cell, suppose this is the diploid oogonium, it will undergo the mitotic process. Before birth of the baby, it will get arrested as a primary oocyte, and then the meiosis 1 and the set of meiosis 2 will be uh, completed when the that baby girl will get mature. Then after puberty, whatever the stage has arrested before birth, it will get complete its uh, cycle of the meiotic process, and then the secondary oocyte will become the mature, and then it will complete its meiotic division, and it will here also it will divide unequally. One will be the smaller one, and one will be the larger one. The larger one will remain as it is. It will work as a female egg, and the smaller one will become the secondary polar body. This polar body will get degenerated and only one single female gamete will remain. This single female gamete will get fertilized with the one male gamete, and the fertilization process will get completed. So the oogenesis process will take place here, as this is the structure of a ovary. This is the cortical region which consists of the different developing Graafian follicles, and this is the medulla region. If you observe this single Graafian follicle, it consists of the number of cells, and this is the egg which is surrounded by these follicle cells. And when during maturation, when it will get ruptured, then this is called as a ovulation process, and this egg or the ruptured egg is released, and it is passes towards the fundus region for the fertilization process. So this is a egg of a hen, and it is the outermost shell membrane, and this is the shell membrane. So I hope that you have understand about the process of gametogenesis, and in this lecture we have discussed about the spermatogenesis and the gametogenesis, and we have also compared what is the main difference between the oogenesis and the spermatogenesis. I hope that you have understand this point very well. Thank you for your patience listening. Thank you so much.